What I've described to this point in terms of inheritance patterns are those that are observed when you have Mendelian conditions. So the monohybrid cross, again, very predictable outcomes through the F1 and F2, as well as a test cross population when you have Mendelian conditions. Same thing with a dihybrid cross, very predictable outcomes in all of those different hybrid generations when you have Mendelian conditions. Same thing for a trihybrid and so on. So what are Mendelian conditions that give rise to those predictable outcomes? So the Mendelian conditions are single genes controlling single traits. The allelic variants that exist for any gene always exhibit a dominant recessive allelic relationship. And if you're analyzing two or more genes within the same set of crosses, is that you always have independent assortment. Another Mendelian condition is the idea of diploidy. So again, we'll touch on that in a future lecture. But what I'm trying to give you again are fundamental Mendelian conditions that give rise to the predictable inheritance patterns that I've described. So again, around 1900, when other scientists, other biologists started to perform genetic analysis for their organism of choice, as I've described previously, for some of those traits in particular organisms that they were analyzing, they observed non-Mendelian inheritance patterns. So again, they knew the outcomes were had an underlying biological basis because the outcomes were predictable through these sets of crosses, analyzing F1s, F2s, F3s, test cross progeny, and so on. So they had to determine, what was different from what Mendel observed that gave rise to these new inheritance patterns, so these non-Mendelian inheritance patterns. So there's actually multiple mechanisms that can give rise to non-Mendelian inheritance patterns. And we're going to talk about one of them in this lecture. We'll be talking about the others in upcoming lectures. And whenever you deviate from Mendelian conditions that I just described, you are going to observe non-Mendelian inheritance patterns. So in other words, there are some traits that are controlled by more than one gene. The final phenotypic expression of the trait or phenotypic outcome is not determined by a single gene, but there's multiple genes that contribute to the final expression of that particular trait. So that's a non-Mendelian condition. In addition, for any given gene, Mendel observed a dominant recessive allelic relationship. Well, there are other types of allelic relationships that can occur, something other than dominance and recessiveness. That's actually what we're going to be talking about in this lecture. In addition to that, you can have the non-Mendelian condition where you don't observe, when you're looking at two or more genes, the inheritance of two or more genes together within the same set of crosses, is that you don't observe independent assortment. So in other words, you see dependent assortment. And basically what that means is that the two genes that you're analyzing, instead of being on different chromosomes within the genome, as we've described previously being responsible for independent assortment, the two genes fall on the same chromosome. So they are what we call linked. Again, so they will segregate together as a unit during a meiotic event because they're on the same chromosome. So they don't exhibit independent assortment. And then lastly, there's another condition. So again, in an earlier lecture, when I was describing the human genome, I identified a unique set of chromosomes that distinguishes males from females, so the sex chromosomes. So in Mendel's peas, there was not a set of sex chromosomes. They were all autosomes. Okay, so again, the flower is bisexual, both male and female reproductive structures. Okay, so if you have an organism, such as mammals, humans, and other organisms as well, for which you have a unique set of chromosomes that distinguish the sexes, and you're analyzing the inheritance of the trait that is conditioned by those genes, you will observe non-Mendelian inheritance patterns. So that's a phenomenon that we call sex linkage. So again, we'll talk about that in a future lecture. So what I want to focus on today is on alleles, looking at alleles and other, a variety of different concepts that relate to an understanding of alleles, different forms of the same gene. So in terms of their allelic relationship and other facets of these alleles as well. So again, the Mendelian condition being that whenever you're looking at two alternative forms of a gene in the heterozygous individual, one allele acts in a dominant manner relative to the other allele, the recessive allele, and therefore, the dominant allele determines the final phenotypic expression within that heterozygous individual. Okay, so this is what happened in the early 1900s. So again, for a horticultural crop, here's two examples here, snapdragon and chrysanthemum. So plant breeders, for these crop materials, this ornamental crop, are interested in understanding the genetics of flower color. And again, the reason for that is that then they could, once you understand the underlying basis for this particular characteristic, you can design breeding programs to develop, newer and better varieties that have, different colors, different flower colors that then presumably would be of some sort of economic value. They could sell this material. Okay, so, a very simple experiment. What would you do to determine the genetic basis for flower color? Well, you look for parental types that are pure breeding for contrasting forms of expression of the flower color. So we'll do, here's carnation up here, pure breeding for white, pure breeding for red, or alternatively for the snapdragon down here, the same thing. You cross red by white and you generate F1 hybrids. And then you analyze the flower color, the phenotype in those F1 hybrids. Well, as it turns out, as you can see here, in both cases, the F1 hybrids exhibit a new phenotype that is not present in either of the two original parental types. The flower color of the hybrid is pink as opposed to red or white. So that right there, in and of itself, is a non-Mendelian observation. You get a new phenotype in the hybrid relative to the two original parents. Okay, so they thought that was interesting. And again, they wanted to determine if there was an underlying genetic cause for that unusual outcome. And so they continued their experiments. And what would they do? Well, they would continue, the Mendelian crossing scheme. They would allow the pink F1 hybrids to self-fertilize and analyze the segregation of flower color among the F2 progeny. So here's an example of that. Again, use the snapdragon as the example. So all of these F1 hybrids, they would self. They'd then, grow out the seed to produce the F2 progeny and then determine their flower color. And what they observed, again, was something that was non-Mendelian. In an F2 population, for this particular trait, the segregation pattern was 1/4 white flowered, 1/4 red flowered, and a half of the population, 2/4, being pink. So a 1 to 2 to 1 phenotypic segregation pattern. Mendel never observed that. Okay, so they, as I said, they could repeat this cross over and over again. So they knew that there was some sort of an underlying biological basis for inheritance of this trait, but it was clearly non-Mendelian. So what they did is they looked at that F2 population, that phenotypic segregation ratio, and they recognized the fact that it was equivalent to a Mendelian genotypic segregation ratio for an F2 population from a monohybrid cross. Okay, so again, remember in Mendel's F2 populations for a single gene trait is that genotypically the progeny would segregate 1/4 that are homozygous dominant, 1/4 that are homozygous recessive and 2/4 that are heterozygous. So now we're just seeing that segregation ratio at a phenotypic level instead of a genotypic level. So they said, okay, what that means is that flower color in Snapdragon is determined by a single gene. There's two allelic alternatives. However, those two allelic alternatives don't exhibit a dominant recessive allelic relationship, but instead they exhibit a new relationship that they call incomplete or partial dominance. So what that means is with that particular allelic relationship, you get an intermediate phenotype in the heterozygous individuals. So again, if you mix red with white, you'll get pink. So it's an intermediate, sort of a blended, it's a new phenotype, but it's a blended phenotype between the two original parental types. So to confirm that, what they would do is that based on that model, the prediction is these F1 hybrids are heterozygous, the red allele and the white allele. So they would perform, a test cross. They would cross to the homozygous white in this example here. So you have the heterozygous individual, you cross to homozygous white, little r would be the underlying genotype, or little r representing the white allele. And what they predicted, and then what they actually observed, was a 1-to-1 segregation ratio. So again, that outcome supports the model of a single gene trait, two allelic alternatives exhibiting this non-Mendelian allelic relationship. They could do the same thing. They could take the F1 hybrid pink flowered plants and cross with pure breeding or homozygous red, the other parent. That would be big R, so homozygous for the big R allele. And again, in that case, they produced progeny that would segregate 1-to-1 for red to white. No, I'm sorry, red to pink. Okay, so again, all of that data supports their model. So in this analysis, they identified a new allelic relationship that was non-Mendelian that gave rise to a non-Mendelian inheritance pattern, a non-Mendelian segregation ratio in that F2 population. Okay, so other investigators, researchers, biologists, again, were doing the same sort of analysis with other organisms as well, same sort of genetic analysis, and they identified other or additional allelic relationships that are non-Mendelian. So here's one example of another, a third type of allelic relationship. So this is in white clover. So generally considered a weed. So, weed scientists are interested in the genetic makeup of the weeds, which would allow them to combat the weed, or, develop strategies for killing off weeds in whatever situation you want to do that. So one of the things they performed genetic analysis for was the characteristic of a leaf coloration pattern. So they had different strains of white clover that exhibited different coloration patterns of the leaves. And so here's one pure breeding type that they identified that exhibited what's called a chevron pattern. So again, this is a green leaf, a green with the 3 leaflets, a green background, and then superimposed on that green background is this chevron pattern. So they had pure breeding varieties that looked like that. Another pure breeding variety that looked like this, so phenotypically different. This is what's called an inner light spot pattern or an oval pattern. So again, what you can see are green leaflets of this individual leaf, and then at the base of each leaflet is a white spot. Okay, so 2 different phenotypic expressions for this given trait. So you want to work out the genetics, so what do? You hybridize them. You analyze the phenotype of the F1, you self the F1, you analyze the segregation pattern among the F2 progeny, and so on. Classic Mendelian strategy for genetic analysis to determine the genetic basis for a given trait. So this is what they saw when they hybridized these two pure breeding parental types. Okay. So actually, in this case, it looks like maybe a new phenotype, um, similar to what I described previously. But actually, what it is you can see co-expression of both parental types in the F1 hybrid. So you can see the oval pattern clearly, or the inner light spot, and then superimposed on top of that is the chevron pattern. So you can see both parental phenotypes in the F1 hybrid. So they said, okay, well that's clearly a non-Mendelian inheritance pattern. So what's going on? So what they would do is they would self the F1 hybrids and look for segregation among the F2 progeny, just like I described for the flower color in the previous example. And what they saw is that among the F2 progeny, 1/4 exhibited the chevron pattern, 2/4 exhibited the F1 hybrid pattern, and 1/4 exhibited the oval pattern. So a 1 to 2 to 1 phenotypic segregation ratio again. So they built a model as it's a mod it's that 1 to 2 to 1 phenotypic segregation ratio is equivalent to a Mendelian genotypic segregation ratio in the F2 population of a monohybrid cross. Okay, so they built a model, single gene trait, two allelic alternatives. However, in this case, we have the non-Mendelian condition of an allelic relationship that is something other than dominance and recessiveness. And the difference from incomplete dominance, in incomplete dominance in the heterozygous condition, you produce a new phenotype. So red by white gives you pink. It's a blended phenotype of the two parents. But in this case, you can see equivalent expression of both parental types. So this is an allelic relationship that we call codominance. So it's just a subtle distinction, but there is a difference. Okay, so both when you have alleles that are incompletely dominant or partially dominant or codominant, they both distort the Mendelian phenotypic segregation ratio in the same way among the F2 progeny, but the relationship is different. It depends on how the phenotype is expressed in the hybrid material as to whether you decide that you have a codominant allelic relationship or if you have an incomplete or partially dominant allelic relationship. And sometimes it's confusing. And I'll give you an example of that. So here's an example. Even in genetics textbooks, they confuse this sometimes. So so here's coat color in horses. So again, that's for horse breeders, that would be a trait or a characteristic that you'd want to determine the underlying genetic basis for. So what we're looking at here is what's called a blue roan horse. And so the blue roan has this gray coloration pattern. Okay. So how did you produce this blue roan? What they did is they crossed a pure breeding black horse with a pure breeding white horse, and they generated a gray horse. If you intercross two blue roans to generate a segregating F2 population, they would segregate 1 to 2 to 1 of black to blue roan to white. So again, that seems to be following an inheritance pattern similar to the flower color in Snapdragon. Gray would be sort of an intermediate between black and white. So you would conclude that you have incompletely dominant alleles. Okay, but in reality, if you get up real close to this horse and you look at individual hairs of the fur, what you see is both black and white. So in reality, what you're seeing here is a codominant expression of both allelic alternatives within the heterozygous individuals. Okay, so this is what I mean. You can have this confusion about this particular allelic relationship, whether it's codominance or whether it's incomplete or partial dominance. Okay, so here's another example of codominance. And so the reason I give this example is a little bit different from all of the previous examples that I've been describing in terms of phenotypic outcomes produced from particular alleles at genes in different organisms. So all of the previous examples that I've been using refer to a phenotype that is observed at an organismal level. But you can observe phenotypes at other levels of observation. So in other words, at a cellular level, at a biochemical level, and so on. So here's an example of a cellular level phenotype. So blood group typing in humans. And what I should say is that, for these types of cellular or biochemical type phenotypes that are analyzed through genetic analysis, is that it's actually more common at this level of observation to observe codominant allelic relationships, as opposed to either dominant recessive or incomplete or partial dominance. So here's the example. This is the MN blood typing system. There's an L gene within the human genome on whatever chromosome. There's two allelic alternatives. There's L superscript M and L superscript N, producing two different phenotypic types, M and N blood type. Okay, so for these two alleles, well, the first thing I should say is the idea that now what you're seeing here is that we're representing the alleles in a slightly different manner than what I've done previously. We're using superscripts as opposed to uppercase and lowercase letters. That's not uncommon. So you should never get confused when you're looking at how particular alleles are symbolically represented. Okay, there are many different ways that you can do that, and we'll talk more about that as we go along. In any case, for this particular example, this is the allelic designation for these two alleles at this L gene. So what that means with those two different allelic alternatives, there's three different genotypes that can exist within any individual. You can be homozygous L superscript M, you can be homozygous L superscript N, or you can be heterozygous. So again, if you're homozygous for either M or N, then what does that mean in terms of your blood type? Well, you're either phenotypically M blood type or you're phenotypically N. What about the heterozygote? Well, for the heterozygote, what you have is a new phenotype, but the new phenotype is actually co-expression of both of the original parental types, M and N. So this is a codominant allelic relationship. Okay. So this is, as I said, is very common when you're analyzing either cellular level or biochemical level phenotypes. Okay, so just to sort of expand on that concept, then, is that here's another example of a biochemical phenotype in humans. Again, another blood typing system that I'm sure most of you are familiar with, the ABO system. So again, this is the same idea, is that there's a gene, the I gene, within the human genome that produces an antigen on the surface of red blood cells that is then detectable through an antibody test that allows you to then phenotype the blood type for a given individual. So what's unique here relative to all previous examples that I've discussed is that for this I gene, there are three alleles. So the idea is that, all previous examples, I've always indicated two allelic alternatives existing for any given gene, and then they exhibit a particular relationship in the heterozygous condition. However, in reality, what you find is that for most genes in the genome of any organism, is that there's always going to be more than two alleles. There are some genes for which you can identify, more than 100 different allelic alternatives for that single gene. So that's the reality. So this is the first example of that. In this case, for the I gene, there are three alleles. So you have the I superscript A, which produces the A antigen, which gives, in a homozygous condition, would produce the A blood type. You have the I superscript B, which produces the B antigen, which would produce a B phenotype. And then you have the O, I superscript O allele, sometimes designated with a lowercase I as opposed to I superscript O, in which you produce no antigen and therefore in a homozygous condition produces the O blood type. So three different alleles at the same gene. So the question is what are the allelic relationships within this allelic series? This would be described as an allelic series. So, are they all dominant and recessive relative to each other? Are they all co-dominant relative to each other? Are they all incomplete or partially dominant relative to each other? What's the case? So the only way you can determine an allelic relationship is by looking at heterozygous individuals. So to determine a particular allelic relationship between any two alleles within this series, you have to analyze the heterozygous individuals. Okay. So that's what we have here. So as I said, the homozygous genotypes are going to produce the appropriate phenotype, your homozygous I superscript A, the A phenotype, homozygous for IB, I superscript B, a B phenotype, homozygous for O, the O phenotype, and so on. What about the heterozygous? So if you're heterozygous for A and O, what is the phenotype of that particular individual? Well, when you score them through this, this antibody test, this biochemical test, their phenotype is A. So what does that tell you? It tells you that the A allele is dominant to the O allele. If you're heterozygous for I superscript B and I superscript O, again, what would be the phenotype scored for that particular individual? It would be B. So again, that tells you that B is dominant to O. So O is a recessive allele in each of those two cases. Okay, so and the A and the B are the dominant alleles. So what about A relative to B? So if you're heterozygous for I superscript A and I superscript B, what's your phenotype? Well, in that case, your phenotype is the blood type of AB. So what you're seeing in those heterozygous individuals is equivalent expression of both of the original parental types. So you would describe that allelic relationship as one of codominance. So the idea is that when you have multiple alleles, more than two alleles at any given gene, you can have different types of allelic relationships within that allelic series. It depends on who you're comparing to who. So again, in the one case, for this example, the I superscript A allele is dominant relative to O, but the same allele is codominant relative to B, to the I superscript B, but it's the same allele. Okay, so again, the designation of whether a particular allele is dominant, recessive, codominant, incomplete, or partially dominant depends on who you're comparing it to. So it's actually a relative designation. And actually, it doesn't really mean that much. It only makes sense in a particular context when you designate an allele as being dominant, recessive, or otherwise. Okay, so you can confirm, these allelic relationships that I just described to you through genetic crosses. So again, if you have two individuals that are heterozygous for A and O, and we've described the allelic relationship as being dominant and recessive, how should the progeny segregate? Well, 3/4 should be A, phenotypically, and 1/4 should be O. And that's exactly what you would observe, and that's a segregation ratio that you'd expect for, the phenotypic segregation ratio you'd expect for dominant and recessive alleles. Same thing for B. If you cross two heterozygotes, the predicted phenotypic outcome would be 3-to-1 of B to O, and that's exactly what you would observe. So again, Mendelian with that dominant recessive allelic relationship. However, if you cross two heterozygotes with the AB phenotype, their progeny would segregate 1-to-2-to-1, exactly what you would predict for codominant alleles. Okay? So again, that confirms the idea that those two alleles, or the allelic relationship between those two alleles, is one of codominance. Okay, so here's another example of an allelic series. In this case, there are 4 different alleles at a single gene. So this is another mammal. We used the horse coat color earlier. So now we're looking at coat color in rabbits. There's a gene that determines that particular phenotype. It's called the C gene. There's 4 different alleles. There's C superscript plus. Again, we're using, these superscripts to designate and distinguish between individual alleles instead of uppercase and lowercase. So this is the brown allele. Homozygotes would look like this with a brown coat of fur. There's a second allele. It's called the chinchilla allele, C superscript CH, produces a gray-coated animal. Then you have the Himalayan allele. Homozygotes would look like this. Phenotypically, this is a Himalayan phenotype, and what you see is that the animal is pigmented, or the fur on the animal is pigmented only at the extremities, the cooler portions of the body. The feet or the paws, the tail, around the mouth and nose, and the ear. So you only get pigment accumulating in the hairs of the fur in these cooler portions of the body. And then lastly, you have a 4th allele, which is an albino allele, little c, where you get, a white coat of fur, no pigment. So again, the question is, now with these 4 alleles within this allelic series, what are the relationships? Well, again, the only way you can determine an allelic relationship is if you look at heterozygous individuals. So that's, you make all of the different combinations, and then you derive particular allelic relationships. So in this case, what you can see is any of the heterozygotes with the brown allele, the C superscript plus allele, are going to produce a brown-coated hybrid. So that tells you that brown is dominant relative to the other 3 alleles. The other 3 alleles are recessive to brown. Okay, but what about these heterozygous combinations? This is chinchilla and Himalayan, or chinchilla and albino. In that case, for these heterozygotes, you produce a gray-coated animal. So again, the chinchilla pattern. So that tells you that the chinchilla allele is dominant relative to the Himalayan and the albino allele. But that's the same chinchilla allele that I just described as being recessive relative to brown. So it's the same allele. In one context, it's acting as a recessive, and in another context, it's acting as a dominant. So this is the idea, the concept that these allelic relationships don't really mean anything except in a particular context. Depends on who you're comparing to who. Okay, so they're not fixed designations. So here's another example. If you're heterozygous for the Himalayan and the albino, you get a Himalayan pattern. But that's just, so the Himalayan would be dominant, the albino recessive. So again, that's the same Himalayan allele that I described up here as being recessive, and up here as being recessive as well. So it's a relative designation when you call an allele either dominant, recessive, codominant, or whatever. And then lastly, the only time you'll produce an albino rabbit is if you're homozygous. So again, that means that the albino allele is always recessive. Certain in this series of four, recessive to the other three alleles. Okay, so going back to just give you one other example of this idea of an allelic series and different allelic relationships that can occur within a given allelic series is the white clover. So previously we talked about codominance between that oval pattern and the chevron pattern. So actually here's the oval pattern here, and here's the chevron pattern here. Okay, but actually for this particular gene in white clover, there's 7 different alleles. That's what's being shown in this column over here. These are all the homozygous genotypes and phenotypes that result for those 7 different alleles. And then everything else represents different heterozygous combinations. So for instance, here's the inner light spot, the chevron, here's the F1 heterozygote, the same as that I showed you earlier. So that we concluded that exhibits codominance because you can see both parental types in the hybrid individual. Okay. So, however, if just to use one example, is that if you were to take the oval pattern allele, this one here, V superscript F, and you compare it relative to this one, V superscript L, this is the result. This is the hybrid here. And it looks to me like that's all you see is the oval pattern. So again, in this case, so in one context, the V superscript F allele is codominant relative to V superscript H, but it's dominant relative to V superscript L. And you can go and you can look at these other patterns and try to determine, particular allelic relationships, but they're variable. It depends again on who you're comparing to who in the heterozygous state as to whether or not they're, you describe a particular allele as dominant, recessive, or whatever. Okay. Okay, so I've described now 3 different types of alleles, or allelic relationships. So, the Mendelian dominant recessive, we talked about codominance, we talked about incomplete or partial dominance. And what we saw when you had these new relationships, these non-Mendelian allelic relationships, is that you see non-Mendelian inheritance patterns. You generate phenotypic segregation patterns among segregating populations that are non-Mendelian. So for codominant alleles and incomplete or partially dominant alleles, we saw a 1 to 2 to 1 phenotypic segregation ratio in the F2 population because of that non-Mendelian allelic relationship. And then we talked about this other idea of allelic relationships being a relative designation, and then also introduced the concept of allelic series. So again, the idea that in the real world is almost always you'll find more than 2 alleles for any given gene. Okay, so I want to describe another type of allelic relationship now, and this is a relationship that gives rise to lethality. So these are what are called lethal alleles, disease-causing alleles, uh, uh, alleles at particular genes that alter developmental programs. So it doesn't result in a live birth, and so embryonic lethals and so on. Okay, so lethal alleles again can exist in 2 forms, either a dominant form or a recessive form. So what I want to first point out is that the most common type of lethal allele that's observed in nature are the recessive alleles, recessive lethal alleles. And there's a reason for that. So if you think about it a second, so if you have a normal, wild type functional form of a gene, so one allelic form that gives rise to a healthy individual, then you have a recessive form of that same gene that gives rise to lethality, right? Then in a population of organisms, 3 different genotypes can exist. You can be homozygous for big A, you can be heterozygous, or you could be homozygous for little a. So if within that population the capital A allele is a dominant lethal, what does that mean? It means 2 of the 3 potential genotypes within that population are going to die, right? So those are lethal genotypes. If it's acting as a dominant lethal, both of those genotypes, either homozygous big A or heterozygous big A little a, are lethal conditions. And if that lethality kicks in prior to reproductive maturity, then that lethal allele, that dominant lethal, will never be transmitted to the next generation. So in contrast to that, if you're dealing with a recessive lethal, the only genotype of these 3 possibilities that actually is lethal would be homozygous recessive. So if you're a heterozygote, because that's a recessive lethal, that would be a viable individual. And they could survive to reproductive maturity, they could reproduce, and they could transmit that recessive lethal allele to the next generation. So this is the reasoning as to why it's more common to observe recessive lethals in populations of organisms as opposed to dominant lethals. And it's not to say that dominant lethals don't occur, it's just that they're rare. And the way you can propagate a dominant lethal allele is that if the lethality kicks in after reproductive maturity, so then they still have the opportunity or the chance to transmit that dominant lethal allele to the next generation. Okay, so in any event, I'm going to describe an example of a recessive lethal and show you how it produces non-Mendelian phenotypic segregation ratios in segregating populations. I'm actually going to describe the very first instance for the characterization of a recessive lethal allele. Okay, so again, as I said previously, the early 1900s, different investigators were trying to develop different organisms for, as a model system for performing genetic analysis. And so certainly in a research lab, the mouse became an important model system. Again, presumably any sort of genetic information we can derive from the mouse would be applicable to the human condition, which is certainly true, even nowadays for all types of, biological studies. So what they had in this case is, what they do is they maintain colonies of the mice, and these are pure colonies, genetically uniform. Okay, and so the majority, and this is true, early on as well, is that the majority of these mice, these wild type mice, pure breeding and so on, look like this. So they have a particular coat color that we call agouti. Okay. So in one of these colonies of pure breeding agouti mice, they recovered a mouse that looked like this, that had a yellow coat of fur. Okay. So they said, great, we now have a contrasting form of expression for that trait, so we can do genetic analysis. Okay, so that's what you need. You need to have 2 parental types that contrast in their form of expression for a given trait. So what they did is they intercrossed these 2 mice. They had this single mutant or this single variant. They could then cross to a pure breeding agouti mouse. And so that's what they did. And when they did that, they produced, multiple litters, multiple progeny that segregated in a very characteristic way. They segregated one half that were agouti and one half that were yellow. Okay, so a 1-to-1 segregation ratio. So they were able to at least propose a tentative model for the genetic control of coat color in the mouse based on that single observation. So the model that they propose is that it's a single gene trait with dominant recessive alleles. Okay. And so then the question is well, which is dominant and which is recessive, yellow or agouti? So the reasoning is that we knew that, or they knew that the agouti mice were pure breeding. So they must be the homozygotes. And then the yellow mouse must be the heterozygote. And so what that then tells you is that yellow is dominant to agouti. So again, their model was a single gene, say the A gene for coat color. There was an A superscript Y allele that was the dominant allele for yellow color, and then the A allele, capital A, was the recessive allele, which conditions agouti. So that's their model. Okay, so that's just based on very preliminary data. So you want to get more data to support the model. So what could they do? Well, now they had a bunch of yellow mice, both male and female, so they could intercross them to produce. Again, remember their model says that individual, those yellow mice are heterozygous. So crossing heterozygous by heterozygous, the prediction would be in the F2 progeny or the F2 population is that they should segregate 3/4 that are yellow and 1/4 that are agouti. Okay, but they didn't see that. Instead, what they saw very consistently through multiple crosses was that 2/3 were yellow and 1/3 were agouti. Okay, so again, it was very repeatable outcome, from multiple crosses. So they knew that, there was an underlying biological basis, but they had to explain it. So they said, okay, well, it's still a single gene trait. You have dominant recessive alleles, yellow being dominant to agouti, but in addition, that yellow allele also acted as a recessive lethal. So in other words, if you were homozygous for the yellow allele, that would be a lethal condition. And that therefore would give rise to a 2-to-1 segregation ratio in that F2 population. So here's the sort of the Punnett square that would predict the outcome among the F2 progeny. You can see that the yellow mice, the 2/3 are going to be heterozygous, A superscript Y and A, and then the agouti mice are going to be homozygous for A. And then when you're homozygous for the yellow allele, that's a lethal condition, which then produces the 2-to-1 phenotypic segregation ratio of yellow to agouti that they observed. Okay, so there's a couple of things that are going on here. So first of all, demonstrating the idea of recessive lethality and how it distorts the expected Mendelian ratio of 3-to-1 because of the recessive lethal characteristics of that yellow allele. You produce a 2-to-1 segregation ratio, phenotypic segregation ratio. The other thing that's going on is a new concept that I'm introducing now. This is something that's called pleiotropy. And again, pleiotropy is fairly common. So this is the idea that a single gene can actually affect multiple traits or multiple phenotypes. Okay, so we have the single gene, the coat color gene, that actually is affecting two phenotypes. It's affecting coat color, but it's also affecting viability, right? And then the third concept that's being demonstrated here is that the relative nature of allelic relationships that I just discussed. So the idea is that that A superscript Y allele is acting as a dominant towards the coat color phenotype, but it's acting as a recessive towards the viability phenotype, but it's the same allele. So there's a lot of things that are going on, in this particular example that you should be familiar with, these different concepts. The concept of pleiotropy, the concept of how recessive alleles distort Mendelian phenotypic segregation ratios, and the idea of the relative nature of allelic relationships. So there are other examples of this, the pleiotropy, the recessive lethals, and the relative nature of these allelic relationships. This is another example in chickens. So it's a gene, the P gene, that affects how the legs develop and form on the animal. So it's a single gene, the P gene. There are two allelic alternatives. We'll just designate lowercase p, giving rise to normal formation of legs on the animal, controlling that particular developmental process. And then you have an allelic alternative we'll call big P or capital P that produces short or stunted, deformed legs on the animal. Okay, so animals that have these malformed legs are what are called creepers. Okay. So the idea is that you can never recover a pure breeding creeper. So what that suggests is, they're heterozygous. And what that would also suggest is that the creeper phenotype is dominant to, the normal leg phenotype. Okay. So the first point is that you can never recover a pure breeding creeper, suggesting that they're heterozygous, and then you draw those conclusions. So what happens if you cross two creepers, two heterozygous individuals? Okay, well, the progeny will segregate 2-to-1, 2/4 that are creepers and 1/4 that are normal leg formation. So again, what does that 2-to-1 phenotypic segregation ratio suggest? It suggests that you're dealing with a recessive allele. And in this particular case, if you're homozygous for the creeper allele, as shown in this Punnett square here, that's a lethal condition. So it's an example that's perfectly analogous to the previous that I just described. Okay, so again, it's pleiotropic, and the two phenotypes are affected. In this case, how the legs form, as well as viability, and the creeper allele in this case is acting as a dominant towards the one phenotype, how the legs form, and it's acting as a recessive in terms of viability. Okay, so there are other examples of this. There are many examples. So here's another one in the cat. There's a breed of cat that's called a Manx cat. There's a T gene. The T gene affects tail development. So here's the Manx cat without a, most cats have a tail. This one is absent the tail. That's what makes them sometimes a unique breed that people pay money for. So again, it's the same idea, is that, it's a single gene trait. There's two alleles, big T and little t. The heterozygous individuals will produce no tail. So that tells you that the Manx phenotype, the absence of the tail, is dominant, and the presence of the tail is a recessive. However, if you take two Manx cats and you intercross them, their progeny will segregate 2-to-1 of tailless to tailed, and you can never recover a pure breeding Manx cat. So what does that tell you in terms of the viability phenotype? The T allele is acting as a recessive lethal. So it's exactly the same as the previous examples that I've discussed for all of these different, demonstrating all of these different concepts. Okay, so just to summarize these different points that I've tried to emphasize, in this discussion about alleles and having allelic relationships that are something other than what Mendel observed. So we talked about codominance, we talked about incomplete or partial dominance, we talked about lethal alleles, predominantly, or the predominant type that you're going to observe are going to be the recessive lethals. That doesn't mean you can't have in populations dominant lethal alleles occurring. And when you do have these non-Mendelian allelic relationships, when you produce segregating populations, and the examples I've used here, F2 populations, is you're going to observe non-Mendelian phenotypic segregation ratios. So the 1 to 2 to 1 suggests that you're dealing with codominant or incomplete or partially dominant alleles. The difference between the two is based on the nature of the phenotype that is being expressed in the heterozygous individuals. Again, with codominant alleles, you see equal expression. You can see both parental types in the heterozygous individual. For incomplete or partial dominance, you get a new phenotype. You get a blended phenotype. So red by white produces pink, black by white produces gray, that sort of thing. Okay, so and then the other non-Mendelian relationship or allelic relationship produces, when you have recessive lethal alleles, produces a 2 to 1 phenotypic segregation ratio in an F2 population. So again, another distorted Mendelian ratio due to that non-Mendelian allelic relationship. Okay, the other concept that I introduced here is the idea of an allelic series, which is actually, much more common. Like I said earlier, there are certain genes in certain organisms that you can identify more than 100 different allelic alternatives existing for any given gene. The other concept that I try to describe here is this idea of the relative nature of allelic relationships. When you say something is dominant, recessive, or otherwise, it really it only makes sense in a particular context. Depends on who you're comparing to who. And then the last point that I tried to describe in this in this discussion is the idea that students in past years, when they're working problems, they get confused when the symbolic designations for alleles are given. And so, when you start to expand beyond, having two allelic alternatives, one being dominant, one being recessive, the dominant represented as a capital letter or an uppercase letter, the recessive allele is a lowercase letter. So, there are many different ways to symbolically represent allelic alternatives for a gene. So you can use superscripts, as I gave a couple of examples of that. You can use subscripts. You can use multiple letters. You can use combinations of letters and numbers. There's all different types of things that you can do. And so you shouldn't get hung up, or you shouldn't get confused when alleles are represented in these slightly more, these alternative forms. So a worst case, say you had an allele that was designated, ABC. I mean, you can change that to just big A, when you're working the problem. Or the allelic alternative being little ABC, you could just change it to little a to simplify it to something that may be a little bit more familiar to you. But you shouldn't get hung up on how allelic uh, uh, differences at a given gene are represented symbolically.